Howdy, my name is Seth Murray. I'm a professor in the Department of Soil and Crop Sciences at Texas A&M University and Texas A&M AgriLife Research, and I'm a maize breeder, a corn breeder. And I want to thank you and the soybean community for inviting me to come speak to you today. I'm not completely familiar with everybody in the soybean community, so hopefully I targeted at the right audience, and please feel free to ask me questions if I didn't. So I'm going to talk about transdisciplinary plant phenomics and phenotyping for breeding and genetics. And the first thing I would like to remind everybody is how genomics has worked. In 1986, we saw the first QTL linkage map. And at that point, the idea was that we would map loci, and those loci would be useful in some type of marker-assisted selection, at least within those families, if not beyond. That generated a lot of knowledge. It's generated a lot of publications. But at least in the case of maize, we can't point to many examples where it generated crop improvement. Maybe soybeans a little bit different. In 2005, we saw a switch to association mapping. Multiple reasons that don't need to be discussed here, but the goal was that we would probably discover more robust loci across more diverse populations. But again, it generated a lot of knowledge, generated a lot of publications, a lot of students trained, but the change in crop improvement still is hard to point to. In 2009, we started seeing a shift to genomic selection or genome-wide selection. And I'm going to assume this audience knows what that is, but the change has been rapid, at least within industry and within some breeding programs of adopting this. And most people have nothing but very positive things to say about it. Now, unlike the previous two techniques, it has generated some knowledge, but it's really hard to publish on a genomic selection model that only applies to your breeding program in your environments and with your marker set. And so it's really not something that can be published widely, but it is something that's very useful for crop improvement and maybe even for graduate student training. But if we look at the bottom, it's important to think about the mindset that's changed over that time. Going from a mindset of discovery or estimation of traits into prediction of the best genotype, right? And we can add value indices in there, and we can really hit breeding goals a little bit better. And as we think about high throughput field phenotyping, we're still really in this first phase. It's only, we're 30 years behind where genomics is. And so what most people are still trying to do is automate routine measurements or routine traits. So plant height is a great example of that, and I'll talk about that today. Or estimate grain yield. And in sorghum and wheat with the panicles up on top, that's easy to do. In cotton, it works beautifully where you can see the bolls. But in corn, where the ears are tucked underneath the leaves, it's not so easy to do. I'm guessing soybeans sort of fall somewhere in the middle. Estimating disease is something that is being done. But what I'm a lot more excited about is finding new signatures of eliteness, having a new tool that we can't just automate from past experience. And so we're already seeing that with temporal growth patterns and in biomass estimates. We're seeing that in spectral signatures, and I'm going to talk about both of those today. But ultimately, where I think we're going is towards phenomic selection. And phenomic selection would mimic genomic selection using phenome-wide markers, maybe that we don't know are important a priori, to select the best and predict the best individual. Now, whatever we discover from any of these, the most important of those phenotypic markers are going to help identify new stress signatures for farmers, and they're also going to identify new phenotypes and mechanisms of biological importance that we can track and investigate for tools like gene editing. Now, I'm going to assume most people have dabbled in UAVs, and I know people like Brian Deers, for instance, is one that comes to mind who's been doing a lot of UAS phenotyping, Aaron Lorenz and a bunch of other folks. And the pathway is fairly complicated. The first step is placing ground control points in the field, still not an exact science. Then flying the UAV to collect pictures, and that seems to be somewhat of the easiest part. Then downloading the images, doing some type of spectral correction for the various light shadows that might occur. Then to ortho rectify and mosaic the images. And this is probably where the most improvement and the most data intensive process is. And what this really is stitching images together, just like you do at Panorama on your camera. And we have to do this because we can't tell what plots are in any individual image. It's only once we stitch those images together can we really see this. And when I think about what Anthony Hurst and Katie Rainey have done, developing software that doesn't require these mosaics, that's really an exciting tool, but we haven't really gotten into that too much yet. We then make structure from motion 3D point clouds so we can identify structure. We then have to identify the plots, separate the ground from the plants, extract the data by the plots, and then we can export it into some type of statistical software. That's the first point where I, as a plant breeder with my training, can actually use the data and get my hands on it. So this whole process has really taken a huge interdisciplinary and transdisciplinary team of remote sensing people, geographers, statisticians, and of course, agricultural and mechanical engineers to do a great job of figuring out how to do this. And now we're to the point where within my lab, some of the graduate students can do most of this work, but we're really looking for tools like Phoenix and Hyphen that are going to help us automate this process. I will point out one of the biggest challenges we had when we first started was identifying plot boundaries or developing shape files to extract information from individual plots and associating that with the type of information or barcodes that I use in my breeding program. So I wrote some script using an A/B line to sort of do this, and Dr. Stephen Anderson, a former graduate student with me who now is a postdoc at the University of Florida, really ran with it and developed a package in R that I think a lot of people have found very useful for doing this. And some software has this built in, but when you're going from working with an engineer who may be working on 3 or 4 different plots to breeding scale of thousands of plots, tools like this are really absolutely critical. So one of our case studies that I'm going to talk about today is the Maize Genomes to Fields project. And I'm assuming that a few of you have heard of it. It is, I think, the largest public sector breeding, cooperative breeding or genomics and phenomics project that there is. There's about 40 investigators from across the country, all growing the same sets of material and hybrids and taking the same traits using a standard operating procedure to help us get at genetics by environment, by management, as well as the main effects in those measurements across a huge gradient of the United States. And so we use the Genomes Fields project just within College Station in a USDA NIFA plant breeding project to really develop tools for UAVs and try and make some discoveries in UAVs and how they might be used in a breeding process, or at least through G2F. And so in this experiment, there were about 240 genotypes, 2 replicates, so 500 plots. And we used 3 treatments. We used optimal, where we used full irrigation, full fertilizer, dry land, which is what most of my farmers in my area would experience, no irrigation, so you reduce the fertilizer, and then heat stress. So we just delayed plant that about a month later, give it full irrigation, and those plants get much taller, but much more heat stressed. Then we used a number of different phenotyping tools from copters to fixed wing aircraft to ground vehicles, and we rotated those throughout the life of this project. But what I really want to point out is 2017 data, and this again is Dr. Stephen Anderson's work with a number of our collaborators, looking at 2 different phenotyping platforms, a tough wing, which is a fixed wing platform, and then a Phantom 3 Pro copter. And that's over the 3 different management that we had, from optimal to dryland to late planted. And what we can see here is the number of height growth curves taken from all these flights. And what's really important and exciting to us was that previously we'd only take height at the terminal time right before harvest. And what we can see is earlier in the season, maybe it's not so clear in these figures, but there's a number of crossover effects. So the plants that are tallest at the end of the season are not necessarily the plants that are tallest at the beginning of the season. And the reason this is important is because the plants at the end of the season in my environment in Texas are highly correlated to yield. And that has to do with stress resistance, we believe. And so now that we can get at earlier height measurements, we can actually find better correlations to grain yield. And so that to me was really exciting. So this was published in the Plant Phenome Journal in 2020. Now, Dr. Anderson took this further, and this is with a population of recombinant inbred lines, where he took height and developed a logistic growth function to actually interpolate height between UAV flights. He then used those estimated flights in each day to do QTL mapping. And what's really exciting here isn't the individual QTLs, which I doubt we're ever going to take the time to validate, let alone use in a breeding program. What's exciting is it showed me that the QTL we typically would measure at the end of the season by measuring height are not the full story. There's a lot of QTL that show up earlier in growth and then are essentially lost, or no longer creating enough variation to be measurable after later in growth. And so that tells me that there's a lot of exciting biology that we can get to through this temporal dissection of growth, and maybe find new loci that would be useful to modify for improving overall yield. And so this was published in Plants Direct just this last year. Now, plant height is, a good tool for me in corn here in Texas. But what we really want to move towards is, additional traits and getting additional information. So a PhD student with me, Natalia Cruzada, using Dale Cope's fixed wing data, collected a total of 532 variables within genomes to fields. And that was 28 phenotypic features from different vegetation indices using both crop masking and not crop masking, and then taking things like the standard deviation of the vegetation index in the plot, or the mean plot vegetation index, and then also doing plant height. And she threw all of this into a model. And what really blew me away and was very exciting was that we were able to predict the top 10% of yielders almost perfectly. Now the R-squared is only 0.48, but we don't really care all that much about predicting this lower material. We want the best varieties. And of course, some of the material from the far north that doesn't belong in Texas showed up pretty well in that. But I challenged Natalia because, flying 15 times is not practical for a breeding program. So could we actually reduce that down to, a few traits or a few flights? And again, blown away, super excited about this. We can still predict the top 10% of yielders. The correlation has dropped, but with only 3 flights. And this is a flight before flowering, during flowering, and then after flowering, but still a month before we would harvest. So this can help us speed up our breeding cycle. And these NGRDI, which is one of the most important vegetation indices in maize, but not necessarily in wheat or soybeans, canopy cover, plot mean, and plot standard deviation. So not necessarily things that I would be looking for if I had a trait-based focus to my breeding program. It does raise this question, though: is this prediction based on something physiological, or is this prediction, like genomic selection, based on some type of relatedness? So our first hint at getting at that was looking at this vegetation indice for a couple lines that we pulled out. And so we're showing the days after planting, and then the NGRDI vegetation index measurement here, and comparing 7 hybrids. So these 3 commercial checks, DeKalb, Terrell, and BH, are presumably unrelated. And then we have essentially half Civ hybrids here, all to a common parent of TX714, and then 4 pretty much unrelated lines in the program. And what you can see here is that there's much more similarity about the trend of the vegetation index for these half Civ hybrids than there is for these various unrelated commercial hybrids. And that to me tells me that we're definitely picking up on relatedness, and we're doing so not by just one measurement, but by looking at that measurement over time and getting many more measurements. We can see that this data is kind of noisy, though. It's kind of hard to interpret. And vegetation indices are subject to when the tassels come out. They have a different color. Windy conditions can lay over the plant. But time and time again, we've seen this noise is actually a big challenge with the UAV phenotyping. Now, going beyond that, the Genomes to Fields project is sort of a genetic study. Does this actually work in a breeding program? And so this is really fresh data. Only have seen this in the last couple of weeks from my PhD student, Alper Adak. And he's extracted 15 different vegetation indices in 100 elite hybrids that we planted in an optimal planting and a late planting. And he looked at flowering time and yield and predicting breeding values using a bunch of different software to go through this whole process. So it's still not a streamlined decision-making tool. And what he did that's really unique is he actually created a mixed model with flight in the mixed model so that we could actually put all the data together from the whole time series. And what I'm showing you here is different vegetation indices on the x-axis from our dryland and our irrigated trial, our delayed and regular optimal planted irrigated trial. The white dots here are the repeatability or heritability had been, a randomly mated population or a Hardy-Weinberg population. And then our R-squareds on these are very high. So the variance components here are these bars. And what we can see is the individual flight is the largest source of variance in making these predictions for yield. We can see, though, that there is a lot of variation still in the pedigree or genotype here, but that it differs from vegetation index to vegetation index. So these heritable vegetation indices can all be potentially used in a model. And so when Alper did this and plotted this out over time, In this case, the yellow is high yielding and the red is low yielding. And we can see that certain vegetation indices clearly separate the high yielding from the low yielding material throughout time. A couple of these ended up being much more stable between the two different experiments, whereas others like this, the canopy height or crop height would be actually very hard to compare between the two plantings. And so finding stable indices that we can measure over many different environments or treatments or managements is really important. Now, when we put this all into a model, he used 3 different types of regression and found that the linear regression was actually performing the poorest. So our root mean squared error and a cross validation was much higher for a regular linear regression. Now, if we use the tools of genomic selection like ElasticNet or Lasso or ridge regression, we see that the root mean squared error is very low. And furthermore, the R-squareds on these models are very high. So they're very predictive. And this is a 10-fold cross validation. Or I'm sorry, 10 repeats of a 60-40 cross validation. So this is really exciting to me. This tells me that we've got a lot of new measurements that we can use in trying to predict yield. And I mentioned phenomic selection. So we've really only done one true phenomic selection experiment so far, and that's using near-infrared spectroscopy. And this was to validate the really exciting results of Rinson et al. That was published in 2018, where they actually showed that near-infrared spectroscopy could predict wheat and poplar yield from the grain or the wood fiber better than genomic markers because it was cheaper and it was just as accurate. We happen to have a GWAS study that we had scanned into NIR, and so we thought we would try a similar approach. And for those of you who aren't familiar with near-infrared spectroscopy, it is based on solid physics. You scan in the grain, you grind it, you send it for chemical analysis, and you develop a partial d-square calibration. And this spectra that I'm showing you here has somewhere in the neighborhood of 2,400 different wave numbers that it's scanning for reflectance values on each sample. And so we can see this. The partial least squares then is used to develop a predictive calibration for samples that were not sent for wet chemistry. So it's a way to scale our phenotypic measurements much greater. Now, one of the things that's hard when we work with engineers is they want to see calibrations like this crude protein. And so what I'm showing you here on the bottom on the x-axis is the actual value of crude protein measured by wet chemistry. And on the y-axis is the calculated or predicted value by near-infrared spectroscopy. So anybody would say that this crude protein value calibration is much better than phosphorus. However, what's really cool is once we put this into our breeding scheme, we actually find that the phosphorus calibration has less overall experimental error and just as much genetic variation explained. And it's important to note the range of variation is almost the same, twice, from 0.23 to 0.5 in phosphorus, and the same, fold in crude protein. So Holly Lane was a very talented master's student with me who since graduated, and she wanted to take on this project. And using traditional partial least squares, she was able to find that the prediction of yield was fairly high. So this is using standard near-infrared spectroscopy. She found that protein was correlated to yield by 0.58. And if we use protein, starch, and oil calibrations, that was correlated to yield by 0.64. But that's still less than what she was finding as optimal in yield prediction. So that tells us there's something beyond just the composition going on here. Now, when she used the full data set that had whole kernel samples, she found a pretty decent correlation, especially over different years and different stresses. And then when we started working with Jose Crosa, he used functional regression, which is a really amazing technique that was much more accurate over all of the samples, particularly when we started looking at applying these calibrations that we developed in a GWAS study to various breeding trials. And you can see here that the correlation ranges from 0.6 to 0.21 of individual unrelated breeding trials. And to me, that's really exciting. It's telling me that we're probably predicting something based on composition, but unlike genomic markers or genomic selection, we can actually extrapolate beyond just the germplasm that we initially scanned in. And so this was published in the Plant Phenome Journal in 2020 by Holly. Really exciting paper. And just yesterday, or just today in the North American Plant Phenotyping meeting, Jeff Endelman's group in Potato at Wisconsin has shown for the 4th time that phenomic selection actually works better than genomic selection. So, I think, I've moved my program away from genomics almost completely at this point because the phenomics is just so much more exciting. So what do I think the future looks like in this? My goal is going to be to extract as many different features as possible. So vegetation indices over time, structural differences, the remote sensing folks talk about texture. Texture isn't something we can measure. It's just something you see in an image. And if you can develop an algorithm to extract it, we can test the heritability and test its predictive ability and use that in trying to predict yield. And then if it turns out that it's very predictive of yield, we can look at that as a way to discover some important new biology. And so just to imagine this in a matrix format, we're going to have some dependent traits of interest like yield and nutrition and ecosystem services that we can develop some type of index for. We're going to have physiological measurements like flowering time, leaf angle, leaf number that we take in the field. We're going to have temporal measurements like height at 60 days, height at 30 days, height at 90 days. And then these random phene features from remote sensing, and then maybe some genomic features of genetic markers. We're going to hopefully put this all into a growth model and develop relationship matrices and use statistical analysis like functional regression and cross-validation to predict the best untested hybrids in untested environments. And that's what I believe the future of plant breeding is going to look like very soon. So I just want to thank everybody in my group. I acknowledged a lot of the people individually, and a lot of these other people helped with data collection are there now. A lot of the Texas A&M USA Unoccupied Aerial Systems and Agriculture team, a number of other collaborators. And most of our funding for this work has come from the USDA NIFA, both as a hatch project and as an AFRI award, from Texas Corn Producers Board, and from Texas AgriLife Research. So thanks a lot for your time, and I'm happy to take any questions if I have any time left.